This is podcast number 50, and all this week we're talking about emergent properties of biological systems. This chapter focuses on cells, and this podcast talks about how cells can start to cooperate together and act more like what we consider a multicellular organism, and then finishes off with talking about slime molds, which has a phase of its life that is unicellular and a phase of its life that is multicellular. And a big theme of this class is that some things are hard to define in biology, like what is life or what is a cell. Um, and this is an example of, what do you consider an animal, or what do you consider a plant, what do you consider fungus? You're going to see that slime mold is hard to classify. So um, we talked about in the last podcast about how bacteria can communicate with each other through quorum sensing. They can also produce biofilms, which are extracellular molecules that form this film, and they act to help the individual bacteria in a community. So this little figurine here is a Roman phalanx, where it's a formation of fighting people where they protect the uh, upper surfaces and the side surfaces, and allows them to invade areas that individuals couldn't invade. So they all have to work together, and they have this emergent property of protection of the group. And bacteria form the same sort of protective layer on their outer surfaces by forming these biofilms, and they only form biofilms when they um, sense other bacteria are around. The biofilms are made up of proteins and carbohydrates that are excreted outside the bacterial cell. They're individual cells here in the red circles, and then the biofilm in the bracket, which is again proteins and carbohydrates that are exported outside the cell and shield all the cells. You're familiar with biofilms. Every time you brush your teeth, you use some scrubbing to get bacterial biofilm off the surface of your teeth. Okay. Um, the biofilm is um, a function of the genotype of the colony, in that different genotypes of different species of bacteria produce different phenotypes of biofilms. So here is a wild type phenotype. Here's the same species but with a mutation where one gene is gone, and then where a different gene is gone. And you can see the biofilm is wildly different in those three situations, even though this is the same species, just two different mutations. Um, Biofilms are not all bad, they can be good. They're used in sewage treatment plants. Here's one where sewage enters here and then is treated through a artificial stream that winds through, and then at the edges and surfaces of this stream there's a biofilm. And that biofilm has layers to it, at least 3 layers, and those layers have different characteristics, different sort of environment, and each of those layers are full of bacteria that have excreted an extracellular component to make this film. To give you an idea that the different layers have different sorts of microenvironments for the bacteria living in those. There's biofilm that was grown on glass in a lab, and then probed for different characteristics at different places in the biofilm. Um, in at position A here, if you go from the surface of the film down to the slide surface, you can see that oxygen is relatively high concentration near the surface, and then low um, dirt at the bottom of the slide. And even at at the bottom of the slide, there would be not enough oxygen for bacteria that need oxygen in order to survive. But there are bacteria that don't need oxygen in order to survive, and they can be anaerobic. If you ever have really mucky water that smells when you step into a lake and you stir up that black sediment, you're stirring up a biofilm at the bottom of that pond or lake that is anaerobic, and they're relying on sulfur as an energy molecule instead of oxygen as an electron acceptor, and that's where you get that stink. Okay, and there's pretty steep gradient of having oxygen to not having oxygen across that biofilm. And then just a small space away, about 100 or microns or so away, the gradient looks really, really different. To show you that biofilms can distribute flow through the biofilm, here's a movie where these little fluorescent beads are labeled and show you that they don't flow randomly through the biofilm. There's little channels, almost like they're going through a river channel as they're going through the biofilm. So it's an environment, is what I'm trying to convince you of. It's a little microenvironment that the bacteria have created for themselves. As biofilms don't stay forever, as they get ripped off the substrate, here's flow ripping a biofilm off of a substrate, they can leave remnants behind. They can establish new biofilms. And so that's sort of the biofilms are an example of bacteria living together as a community, and that community has emergent properties such as different layers of oxygen tension, different flow regimes, and so on. So it almost acts like an ecosystem of bacteria, or almost like a giant bacterial organism made out of multiple cells that has different characteristics of different parts of the organism. Another organism that acts like that, in that it acts like a single cell for part of its life stage, and then acts like almost like an animal for part of its life stage, and then acts sort of like a fungus for parts of its life stage, is the slime mold. And I'm going to just play you a video from the book that introduces the slime mold.
When food is plentiful, the myxomebae are completely independent as they feed and multiply. When their food supply dwindles, their behavior changes remarkably.
So those individual cells you saw there are the individual cells of the slime mold.
When they become crowded, they start moving together from all directions, flowing into growing masses containing thousands of cells. Aggregation starts when one cell spontaneously secretes a pulse of a hormone called cyclic AMP. Nearby cells respond by moving towards the hormone and by emitting their own pulse. Soon, this response becomes widespread and synchronized, creating waves of accelerated movement passing through the migrating cells. These myxomebae are streaming steadily towards a distant aggregation center. As always, there are a few stragglers rushing to catch up. The aggregations of cells round up, and then each turns into a slug called a pseudoplasmodium. These slugs are active, searching the terrain for a suitable place to settle and leaving a trail of slime as they go.
So they're called slugs because they act like a slug, like you would see in your garden, like a garden slug. True slugs are animals. These are not animals, but they certainly act like you expect animals to act.
Remember that each slug consists of thousands of free-living amoebae, which now have organized themselves into a cohesive unit and which synchronize their activity. During its travels, an individual slug may fuse with other slugs it happens to encounter. Meanwhile, a few individual mixamoebae are still visible, moving around on the surface. After this migratory activity, the slug rounds up again, but this time it flattens onto the surface and then grows a thin stalk with a fruiting body on the tip. Throughout these maneuvers, the mixamoebae remain separate individuals. During this final phase of reproductive activity, the behavior and fate of each cell continues to be subordinate to a self-generated pattern. The mixamoebae in different parts of the pseudoplasmodium differentiate into three main types of cells with different functions. Some cells form the base and others the stalk, while the rest migrate up the stalk and turn into spores in the fruiting body. The mature spores are later released.
Okay, I'm not sure if you've ever seen those before. We've seen them in the forest in Tahiti, and these mobile phases can be big, like 2 or 3 feet across. And when they decide to enter this last stage where they're forming the fruiting body, even though each one is in a different individual cell, they specialize and form different, almost like tissue types. So what do you call this thing? Do you call this thing a unicellular organism? You call it multicellular because different parts of its life are in different phases. Well, you could say the same thing about you. You started off as a unicellular organism when one sperm fused with one egg that made one original cell, which then became a multicellular organism, you. And you could call those different life stages, and this you can think of as just an extreme version of that. But what are they? Would you call that thing an animal? Do you call it a plant? Well, probably not a plant. It's not photosynthetic. Um, and is it a fungus? Well, fungi do have some characteristics that, like animals have. And you can see in this tree, they look like they are closer to animals than they are to than animals are to plants. But it's in between, and uh, they are hard to characterize, and we probably need more information before we can say what it is, or maybe it's its own thing. When you saw those slugs moving, this is the group of individual cells that are moving in a coordinated direction. They tend to follow light. And how is it that they follow that light? Well, cyclic AMP was mentioned on the video as a hormone. You've seen it before as a secondary signaling molecule. I prefer that you think of it as secondary signaling molecule because um, uh, for animals anyway, hormones are outside the cell and secondary signaling molecules are inside the cell. Slime molds play by their own rules. But if you look at the wavelength of light and how it's absorbed, this is the container, but it has a peak of absorbance at a 260 nanometers wavelength. That's typical for nucleic acids. And cyclic AMP is a type of nucleic acid. It is the most abundant. There are other nucleic acid molecules in the signal transduction pathway, which is very similar to the signal transduction pathway that we talked about before, where you had epinephrine binding to a receptor and then the G protein activated adenyl cyclase making cyclic AMP from ATP. But cyclic AMP in all these different signaling molecules, GTP, ATP, cyclic AMP is by far the most abundant. And so cyclic AMP is what these slime mold cells are attracted to and moving toward, and they move in like waves. I'll show you a video in here of that. This one, okay? So in waves like that. Probably can't look at that too long without getting hypnotized. The details on how that wave is produced are well known. I don't care so much that you know how the wave is produced, other than that the signaling molecule cyclic AMP, you saw that in advanced quote-unquote organisms, how you respond to fear, for example. You also saw that in these so-called simple organisms, the slime molds, and both are responding to the same molecule, and it can elicit very complex behaviors as it does in you. For this class, I don't care so much about that you know the details about how they get these waves, just that the slime mold is moving by going toward cyclic AMP, and cyclic AMP is produced as a signaling molecule. And it's an example of how you can get, as you saw in that video, individual cells can come together and form all kinds of emergent properties. What looks like seeking behavior, what looks like light-seeking behavior, fusing behavior, transformation, what looks like a multicellular organism with organized tissues. These are emergent properties of just individual cells coming together and using cyclic AMP as a signal.